So good morning all of you and welcome back to Excelsure Academy. So today we are going to start with the other topic called biomolecules. In this biomolecules, we are going to study about 4 different biomolecules, that is carbohydrate, proteins, lipids and nucleic acids. Okay, so first one which we are going to start is amino acids. So this is going to be your first lecture in biomolecules. So before we start, we know that about 99% of the substance of all living things are made up of hydrogen, oxygen, carbon, and nitrogen. So if you see your carbohydrate, your proteins, lipids, everything has got your carbon, hydrogen, and oxygen. And in the case of proteins, you have got the nitrogen also. Now, all these elements combine in various proportions and various proportion in order to various ways to form the biomolecules. So actually right now for your syllabus, it is not there like how the first protein and how the first carbohydrate was synthesized in the nature. So now we know that there are enzymes which are helping in synthesizing of these molecules, okay? But what about that time, like when the original species, when we talk about, when the first cell was discovered, when the first protein, first carbohydrate, and first nucleic acid was formed? So that time no enzymes were present, right? Because we know that enzymes are again proteins. Proteins comes from the RNA and RNA comes from the DNA. So what made the first protein? Because to make RNA and DNA also you need enzymes like RNA polymerase and DNA polymerase. So we'll study all this when we start with the mole bio. Okay, so I'll just give you the introduction that time. So no need to go in detail for that. Okay, so these are defined as the organic molecules present in the organisms. They act as the building blocks of life and they carry out the important physiological process. Okay, so we are going to talk about the, when I say macromolecules, those are proteins, nucleic acids, polysaccharides and lipids. And when we talk about micromolecules, they are the amino acids, fatty acids. Okay, NTPs. What is NTPs? Nucleotide triphosphate, from which nucleic acids are made. Okay. So from biochemistry onwards, you have to study and practice a lot like maths, because so many structures are there, you need to study all those. So the composition of the living tissue. So this was the first experiment which was carried out to understand what all molecules are present in a living tissue. So how they have done this thing? So in order to study the composition of living tissue, various experiments were carried out. So first one, what they have carried out was using the trichloroacetic acid, TCA. In the lab, you must have seen also. This is used to precipitate. Okay. It helps in precipitation. So there are, what they did is, they got 2 types of composition. Of course, one was acid soluble, okay, and one was insoluble. So insoluble means here the precipitate was formed. Okay. Now when they determined like what is present in the acid soluble and the insoluble part, they found that the acid soluble part had the micromolecules. So it means what they are talking about, the amino acids, sugars and nucleotides. Okay. Because they are single, so they are polar in nature and then they are soluble in the acids or any polar solvents. So getting the point. So when these molecules like amino acids becomes proteins, sugars becomes the polysaccharides and NTPs become the nucleic acids. So they become bigger in molecule, bigger in size. So in the whenever you add TCA, what happens? The larger molecules precipitate. They come together and get precipitated. Okay. So what the researchers found that in the acid soluble pool, you got only the micromolecules, that is the building blocks of proteins and sugars and nucleic acids. Okay. Now, what they found in the acid insoluble pool. So it was all the organelles which were present in the cells and the size was more than 10,000 kilodalton. Okay. So that included your proteins, nucleic acids, lipids and polysaccharide. So it's clear, right? Smaller the molecule, it is easy for dissolving. Bigger molecules, they become difficult to dissolve. Okay, so they just get precipitate out. Now when they did the analysis, they found that Of course, the water has to be the maximum in the cytoplasm. Proteins are around 10 to 15%. Now the amount of protein is going to be high over here. Why? Because maximum proteins are all your enzymes, correct? And we know that to carry out any cellular process or any cell signaling, mainly these proteins are involved either in the form of enzymes or receptors or any other proteins like peripheral, integral proteins, everything. So that's why the composition of protein is going to be high after the water. Then we had carbohydrates, lipids, nucleic acid, and ions. Okay. So now let's talk about the amino acids. So first we are going to talk in detail about proteins. After proteins, we'll go to carbohydrates, nucleic acids, and lipids. With this, your biomolecules will get over, and we have to study about the metabolism also, like the synthesis of these amino acids as well as the degradation. Okay. Yes. So let's start with the amino acid. So what are amino acids? They are made up of carbon, hydrogen, nitrogen, and oxygen. And in few cases, you also see that sulfur is present. Now, if you have little bit knowledge of amino acids, can you tell me which amino acids have got sulfur in them? Ranjan, can you tell?
Methionine.
It is?
Methionine.
Methionine, very good. And?
Serine.
Not clear.
Serine.
No, it's not serine.
Methionine.
It is cysteine. Okay. So these are the only two amino acids which has got sulfur in them, methionine and cysteine. Okay. So they are the building blocks of all the proteins, and we know that proteins are the molecular instruments through which genetic information is expressed. What does it mean? So even if you have got in the chromosome, you have got the gene becomes your mRNA, and from mRNA you get your polypeptide chain, right? So when you say the gene is expressed, it means either it got transcribed or it got translated. Okay, so then only we say that the gene is expressing. Now, this is the basic structure of your protein. Sorry, polypeptide, the peptide bond. So you see that it is also referred to as sometimes the substituted methane. Now, why substituted methane? Because we know that methane structure is what? CH4, correct? So it has got all the 4 as hydrogen atom. Now, instead of that, if I say that this is the central atom, I have got one H over here, right? Amino group, carboxylic group, and side chains. What you have to remember is this C is called your alpha carbon. Okay, please remember this is called your alpha carbon. And if you have to number it, so you always number. How do you do the numbering? You have to start with the carbon having the most near electronegative atom. Okay, so in this case, it has got the oxygen. So this is your first carbon. This becomes your second carbon. Okay, just remember this much. This is alpha carbon, but if it is in the number system, then it becomes the second carbon. See, I told you chemistry is needed to study bio. Now, history, this is very simple. It's okay even if you don't remember, because I don't think this question is ever asked. So we know that there are 20 amino acids which is commonly found in proteins naturally occurring. The first one was to discover was asparagine. Okay, so it was asparagine, and the last one was threonine. So how do you name it? ASP or N, threonine THR or T. Okay, so this is the nomenclature, and you have to remember very nicely. You shouldn't get confused. Okay, otherwise the sequence goes wrong. The entire the numericals will go wrong. Okay, so asparagine was first found in the asparagus, and glutamate from the gluten. Tyrosine was first isolated from the cheese. Glycine, and glycine was named because of its sweet taste. Okay. So this is just okay if you just go through and all. It's not that important to remember. Yes. Now from here onwards, you have to remember everything. So classification of amino acids based on the synthesis. So we have got the essential, semi-essential, and the non-essential amino acids. So essential or indispensable amino acids, it cannot be synthesized in the body and it have to be taken in the diet. So what are those? Valine, isoleucine, leucine, lysine, methionine, alanine, threonine, tryptophan. Okay, you have got the phenylalanine. Sorry, it's not alanine, phenylalanine. Now, it's on you how you're going to remember. You can make certain coding or acronyms so that you can remember. This is important. From here onwards, it is important. Now, semi-essential amino acids means they are acting as a growth promoters. So they are not again synthesized enough in the body. So whatever is essential, it means not synthesized in your body, is your essential amino acids. Example, arginine and histidine. Guys, you can stop me, okay, if something is not clear. This is something you have to actually memorize. Then non-essential means you can synthesize in your body. So examples are glycine, tyrosine, proline, cysteine, asparagine, etc. Okay. Now we have got the classification based on the, this is the same thing, based on the chemical nature. Okay. So based on the chemical nature, what does it mean? Whether they are polar, non-polar, acidic or basic. So you have to remember all your 20 amino acids for that. Okay, one second. Yes. So non-polar means no hydroxyl group, no hydrogen bonding capacity. Okay. So here you see glycine, alanine. Glycine has got the structure as COOH, NH3, H and H. Okay. So what do you see over here? That it has got the 2H. So instead of one R group, glycine has got H. Okay. So now can you tell me, is it a chiral carbon or achiral? Yes.
Chiral.
Sure. Is it chiral? Glycine is chiral?
No, ma'am.
Ah,
No, ma'am. Because 2H plus H
yes.
are there.
So achiral, it is a achiral. Chiral means that you should have all the 4 different groups. Okay. But in the case of glycine, it is not. Whereas now in the case of alanine, here you have got CH3. Okay. So this is how you are going to study the structure. Like how I remember now, just remember the basic structure, C, COH, NH3, H, and then just keep on changing the R. So CH3 is your alanine, okay? Then you have got the valine, another CH2 group will be there, leucine, isoleucine, proline. Now proline is having different structure. It has got the cyclic structure is there. Okay. Then phenylalanine, you have got the phenyl ring over there. Now if it is a tyrosine, it becomes like this. The structure is like this. This is of phenylalanine. Okay. Now, if I have to make tyrosine, just add another OH group. It becomes tyrosine. Okay. So this is how you have to study all your structures also. Now, polar means either you should have a OH group or a capacity to donate or accept an electron or make a hydrogen bond. Okay. So that becomes polar. For example, serine, threonine, asparagine, glutamine, cysteine, tyrosine. And then acidic, from the name itself, you know. So they have got the additional COOH group. Now, lysine, arginine, and histidine, they have got the positively charged. So on their R group, and hence they become the basic. Okay. Now, on the basis of the side chain. So this is what Akshita has done as her assignment, beautifully done. So you can see over here. See, she started with alphabetical order. So starting with A, alanine, ALA, A, and the structure is there. Okay. Then arginine. Now, why it is R? Because when you say, it doesn't come out as A, it comes out as R, right? Arginine. So that's why it is R. Now, asparagine. So these are all Greek and Latin words, right? So when they write they write in their language, what they sound is N. The maximum time they sound it as N. That's why it is N. Aspartate, so they write as D. Cysteine, C. Glutamate. So main focus is given on the T, so it becomes E. Okay. Now, glutamine, why it is Q? Because those people write as glutamine. So that's why it is Q. Glycine G, histidine only one is there, so H. Isoleucine I, leucine L. Now lysine is K because so many alphabets were already used. So whatever was left, they started putting the alphabets. Then tyrosine, Ti. So Y it is over here. Okay. Then we have got methionine M, phenylalanine, it's Phe. So that's why it is F. Proline P, serine S, threonine T. Now tryptophan, it is W because they pronounce it as tryptophan. Okay. Then valine V. So that's how you have to remember. If you ask me anytime, I remember very well all the alphabets, and you also have to remember, because in future whenever you have to solve the equations, everything is there. So they don't write the entire name of the amino acid. They just write only these alphabets. Okay. Now this is something very important. So absorption of UV light with the by the aromatic amino acids. So which are the aromatic amino acids? If you know the structure, it is your tryptophan, tyrosine, and phenylalanine. Okay. Now, when using the spectrophotometer, I'll take in the next class, spectrophotometer. When you take the readings, so what you have to do, you can make a 1 mg per ml solution of tryptophan, tyrosine, and phenylalanine. And when you take the scan, okay, when you take the scan, you see that the absorbance is near 280 nanometer. Okay, so phenylalanine doesn't give that much absorbance, but mainly the absorption is because of the tyrosine and the tryptophan. Now, if you ask me, if I take a protein, let us take the example of BSA. Can you tell me which protein it is? BSA? Yes? Anyone? Sandhya, do you know which protein it is? BSA? Commonly used in the lab. It is your, yes, bovine serum albumin. Okay. Please remember, because you are going to start your practical soon, so you should know all these things. So whenever we talk about proteins, and if I say take this much of BSA, you should know that I'm talking about bovine serum albumin. Okay. Now, if I have this protein called BSA, same 1 mg per ml. Okay. Now, if I take the scan of this, how I'm supposed to get the scan? So again, I'll get a scan something like this. Only one peak will be there, okay? And that peak will be somewhere around between 280 to 295 nanometer. Please remember, because your tryptophan gives good absorbance near 295 and tyrosine gives at 280 nanometer. If I take all together in the question, maximum times they assume to be 280 nanometer. Okay. So please remember, whenever you talk about absorbance of protein, we always go for 280 nanometer. And these absorbance is only because of aromatic amino acids, namely tryptophan and tyrosine. Okay, is it clear? This is important. Okay, please remember, this is important to remember. Now, let's talk about cysteine now. So there's something called disulfide bond formation. Now this is formed by the oxidation of two molecules of cysteine. Okay. So if you see the structure over here, the disulfide bonds are formed between the cysteine residues, and it helps in stabilizing the secondary structures of the protein. Okay. So in the further classes, we are going to study about the structural levels of the protein. So primary structure means it is not only the polypeptide chain, what you get after the translation. Okay. Secondary means you are going to have all this disulfide bond formation. Then tertiary, you have got the hydrogen bonding and ionic interactions. Okay. Whereas in the fourth, there is subunit association. For example, hemoglobin. So I know that there are 4 subunits are there. So all those single subunits will be formed, and all those 4 subunits will come together with the help of the hydrogen or ionic bonds. Okay, and then the entire protein is formed. Okay. So we need to study all this in detail with the coming sessions. Okay. So coming back to the cysteine. So you have got 2 cysteine molecules over here. Now see the structure. COO minus and NH3 plus. Okay, so your carboxyl group and your amino group is there. Okay, then only what you have to remember is in the structure cysteine you have got additional group as CH2SH. This is your R group. Okay, now what is happening? The 2 cysteine residues are nearby. Okay, and if it has to form a disulfide bond, it has to lose its hydrogen. Now, losing hydrogen means oxidation. Okay, so that is loss of hydrogen. So what is happening now? So you see that 2 H plus and 2 electrons are lost and the disulfide bond is formed. Okay, now this is cysteine, this is cysteine. Can you see the difference in the spelling? This is pronounced as cysteine, this is pronounced as cysteine. One E is not there. Okay, is it clear to everyone? Pallavi, it is clear?
Yes, ma'am.
Okay, you need to study. Okay, you have to practice all this with the structure formation. Yes, now talking about the uncommon amino acids. So now by next class, okay, I might ask randomly to anyone the nomenclature or the naming convention of any 20 amino acids. Okay, so these were the 20 amino acids which are naturally occurring. They're also called proteinogenic. They are also called proteinogenic. So you shouldn't get confused with the names. Like, what do you mean by proteinogenic? So proteinogenic amino acids means your 20 naturally occurring amino acids. Okay. Now, when you say non-proteinogenic, it means either they are modified or synthetically produced in the lab. Okay. Okay. So now talking about the uncommon amino acids, they also have important functions. Even though they are uncommon, but they are synthesized inside the body itself. For example, 4-hydroxyproline. So what is happening? At the 4th carbon, OH group has been added and they are found in the plant cell wall. And the thing is that you need to remember by heart all this thing. Okay, not the structure, but of course, what are their derivatives and what is their function. So starting with 4-hydroxyproline, it is of course the proline derivative and found in the cell wall. 5-hydroxylysine, okay, it is found in the collagen, which is present in the connective tissue. 6N-methyl-lysine, again present in the myosin. Okay, so can you remember 4-hydroxyproline, 5-hydroxylysine, and 6N-methyl-lysine? And where do you find these? In the plant cell wall, in the collagen, in the myosin. So collagen, you know, it is in the connective tissue. Myosin is your muscle motor protein. It is a muscle motor protein. So today, lot of things are there to remember. Now, carboxyglutamate, they are found in the blood clotting protein called prothrombin. So how many clotting factors are there? Anyone who can tell me? Kirtana, can you tell me how many clotting factors are there?
14.
How much, Ranjan? 40, 40?
14.
13, ma'am.
It is 13 actually, 13. Okay. So among that, it is the one which is acting as a cofactor also, carboxyglutamate, okay, in the prothrombin. So it, and in other certain other proteins where it is going to bind calcium as part of their biological function. Now, the other one is desmosin. Now, why it is said complex? Because of its structure. It is a derivative of 4 lysine residues. Okay, so you can see that till now it was single amino acid, 4-hydroxyproline, 5-hydroxylysine, 6N-methyl-lysine. They were talking about one single amino acid derivative. And in the case of desmosine, it is 4 lysine residues are there, and it is present in the elastin. It is again another fibrous protein. Okay, so elastin. Yes. Next is selenocysteine. So instead of sulfur, you have got selenium. Instead of sulfur, you have got the selenium over here. Okay. Now, those all are created by post-synthetic modification, or otherwise called post-translation modification. That is, once the polypeptide chain has been formed, after that it gets modified. Okay. Now, ornithine and citrulline, this you must have heard if you have read about the urea cycle. So this also we are going to do soon. So in the urea cycle, these are the intermediates. Okay, and it is also found in the biosynthesis of arginine. So anything related to arginine, please remember it is the ornithine and citrulline. Yes. Then we have got the pyrrolysine. It is also used by methanogenic archaea, where they actually produces methane. Okay. Now the speciality is that instead of this is coded by UAG. Actually, in our system, UAG is meant for your stop codon, right? What is the start codon? It is the AUG, right? AUG is your start codon, and UAG is your stop. What are the other codons of stop? Yes, anyone?
UAA.
Huh?
UAA.
UA?
A.
UGA.
Yes, UGA is there. Then see, this is what, even though it is simple, you're having doubts. Okay, so this is your another assignment. Get the your stop codons. Okay, and what are their names? I can tell you it is opal. Remember? So just write it down in your notes about the start and stop codons. So in the case of us, like for example in humans, UAG codes for the stop codon. Whereas in the case of methanogenic archaea, it is coding for the pyrrolysine. Okay. Okay. So moving on to the next. So amino acids, they share a common structural features. So mainly for the amino acid, the structural features, as we already know, this is the structure where you have got the one single carbon having a carboxylic amino group H and a R group. Okay. So as I already said here, this carboxylic will become the first carbon and your alpha carbon will be your second carbon. Okay. Now, if suppose you have got more number of carbons, for example, in the case of lysine. So how many carbons are there? There are 6 carbons are there. Now, if I have to start the naming, like you have to represent when I say that 6N-methyl, what does it mean? 6N-methyl lysine. It means that at the 6th position, okay, over here, you have got the methyl group instead of this. Okay. So this is how you change your structures. Now, how it is done? If it is the Greek letter, then starting from here. So we know that this is your alpha. The next becomes beta, gamma, delta, and epsilon. Now, if it is a number system, then you have to write it as 1, 2, 3, 4, 5, and 6, and so on. If you have got the more number of carbon atoms. Okay. So similarly for the others, for example, in the case of let's say the example of cysteine. Okay, so we know cysteine is like this, SH. So if I have to take the number system 1, 2, and 3. Okay, so this becomes your third carbon with your thiol group. SH is called your thiol group. Now, if I have to talk about the Greek letter, then it is going to be this alpha, and this will become your beta. Okay, clear? This is just to understand what will be there in the question. Now, that was all about the naming. Now we need to talk about their chiral centers, stereoisomers, right? So in the case of glycine only, the R group is H. So this is not used as the standard ones, okay? Because rest all have the chiral carbon atom. Only the glycine doesn't have. Now, because of the tetrahedral arrangement of the bonding orbitals around the alpha carbon, that is the second carbon, the four different groups can occupy two unique spatial arrangements, and thus amino acids have two possible stereoisomers. Since they are non-superimposable, they are called enantiomers. Okay, and they are optically active. What do you mean by optically active? That they can rotate plane polarized light. So are you familiar with all these words? Non-superimposable means, see, if I take like this, my thumb is coming this side and this side. So it means what? These two mirror images are non-superimposable. Okay, so they are called enantiomers, and they are optically active. It means when they are passed through the polarized light, they rotate the plane of the polarized light. Okay, if suppose you have got the, suppose the light is there and it is traveling like this. If it is passing through the enantiomers, what happens? They change their plane. So either it will be, either they will be out of focus like this or like this. Okay, or they can rotate also. Now, so this is the example of alpha, the alanine. So you can see that this is important. Okay, so if you have got H this side and this becomes your L. If you have got the H in the left side, this becomes your D. Okay, now what they have done over here is they have taken glyceraldehyde as their reference. Okay, this will be more clear in the next actually. Yes, if you see over here, so in this case they have taken the Fisher. ML Fisher was the person who actually gave this designation. So he said that if you take all these the glyceraldehyde as a reference, you see that, see over here, CHO aldehyde group is overlapping with your carboxyl group. Okay, and then your H is there, H is there. Similarly, every time your hydroxy group is overlapping with the amino group, and then the different groups over here. Okay, so in the case of alanine, it is CH3. So here R is going to vary for different amino acids. So from this, he said that taking the L-glyceraldehyde as a reference, you name your amino acids. So L-glyceraldehyde means those are going to be the L amino acids. D means it is going to be your D amino acids. Is it clear? Little bit. So once you go through all this thing, it will be more clear. So the absolute configuration, it is developed to specify the absolute configuration of the four substituents of asymmetric carbon atoms. So that is your D and L system. We have got R and S also, right? So if it goes clockwise, it is R. If it goes anti-clockwise, it is S configuration. So yeah. Now, there are certain molecules which are amino acids, but they do not follow the D and L configuration. So we know that there are certain, so all the living system, as I mentioned before, in our system, D sugars are acceptable and L amino acids. But there are certain conditions where you also see D amino acids. Okay, so what are those conditions? For example, in the case of gramicidin, what is gramicidin?
Small pore rhinophore.
Yes. Okay. So it is made up from the mixtures of D and L amino acids. Similarly, you also find in tyrosidin and valinomycin. What about valinomycin? It is dash.
Potassium.
What potassium?
Potassium ionophores.
Correct. Very good. It means you people are studying good. So these compounds disrupt the bacterial cell wall, especially of gram-positive bacteria. So you see, even though we are talking about the different topics now, biomolecules, but in bio, there is nothing different. It's all connected. Okay. So now you know what is gramicidin, what is valinomycin, etc. So in this case, you see D amino acids. Where else do you see the D amino acids? I have mentioned, I guess, once. You also see in the bacterial cell wall. peptidoglycan. So when we do the cell structure, we will study that in the case of peptidoglycan also, you also see D amino acids.
D-alanine.
Yes, correct. So in the peptidoglycan, you see the D-alanine. Very good. Okay, so now we are going to talk about the amino acids which are having non-protein function. So already this we know that your GABA, what is GABA? It stands for gamma aminobutyric acid, act as neurotransmitters. Okay. Then tryptophan, again, it is a neurotransmitter precursor for the serotonin. Then also for the catecholamine neurotransmitters like dopamine, epinephrine, norepinephrine, etc. So what do you see over here? You have got the gamma aminobutyric acid, tryptophan, tyrosine, all these are the precursors for your neurotransmitters. Then phenylalanine, precursor for phenylethylamine and tyrosine in humans. In plants, it's a precursor of various phenylpropanoids and are important in plant metabolism. So when we talk about the secondary metabolites, again from these amino acids only the secondary metabolites are synthesized. Okay. Now glycine is a precursor for porphyrin such as heme. So porphyrin ring is there. Remember the structure. If you see the structure of hemoglobin, you have got the 12 porphyrin rings over here, and in the center you have got the iron Fe plus. Okay, Fe3 plus. The iron moiety is present over here. Similarly, you have got the porphyrin ring structure in the chlorophyll also. So whereas instead of iron, you have got the magnesium over there. Okay. Then arginine is a precursor of nitric oxide. Now, nitric oxide, it acts as what? What is the role played by nitric oxide in your body? It helps in dilating your blood vessels. So once we start the animal physiology, it will be more clear. Okay. So it helps in dilating the blood vessels. Okay. Then ornithine, S-adenosylmethionine are precursors of polyamines. So ornithine we have just discussed now. It was present in the urea cycle, also in the metabolism of arginine, correct? And where do you see SAM? Have you heard of this SAM? Again, it will come in the amino acid metabolism. Okay. It acts as cofactors for many enzymes. Then aspartate, glycine, glutamine are precursors of nucleotides. So there is something called de novo pathway or salvage pathway. Okay. These are your nucleic acid synthesis pathway. So there again, with the help of the nucleic acids, with the help of the amino acids, all your nucleotides are formed, and from the nucleotides, your DNA, RNA is formed. Okay. Then, sorry, I couldn't edit much over here, but you can just go through and just what it is written is that there are certain amino acids which are used as a defense against the herbivores in plants. Okay. So what they're talking about here is that, see, every organism needs to defend themselves, right? So suppose plants are there. So what these herbivores are doing, they go and feed on the leaves of the plants. So the plants will produce certain secondary metabolites or non-standard amino acids in such a way that they are causing toxic reactions to the herbivores. Okay, this is one example given over here. So it causes to the predators such as insects illness in people if sometimes of the legumes are eaten without processing. So do you know about the rajma bean? The kidney bean we say, right? We eat the rajma. So if it is not cooked properly and if you're eating like this only, so you will get all the diarrhea because the protein which are present in that called lectin is going to cause diarrhea in your system. So that's why the rajma, we say rajma chawal is very tasty and all, but if the rajma is not cooked properly, you get all the gastric problems. This is one example. The another example I can give is about the cabbage. So we eat raw cabbage as our salad, right? So if you eat more, just imagine you're eating around half of the kg of cabbage without processing, just eating raw, then also you get all the problems of the gastric. So that's why in salad we are just having 1 or 2 thin slices of cabbage, right, in the burgers and all. But if you eat directly, then you might have the problems. So all these problems are because of these amino acids which cause the problems in the humans. Okay. Yes. Now, in the case of industry, they are used as the animal feed supply. So all your soya bean, once the oil has been taken, whatever is left, okay, sometimes we are eating as the soya bean chunks. If it is not being sold out, then they are given to the animal feed as the source of these amino acids, okay. Then they're also used to chelate the cations in order to improve the absorption of minerals from the supplements. It is required to improve the health and production of these animals. So what they're doing is like, see, it's costly to produce crop differently for the animals. Okay, so it's always better that whatever has been harvested and processed, whatever is left, just add little bit of supplements to that and provide to the feed as feed to the cattle. So in the case of industry, they do such things. Yes. So then again, glutamic acid, this you must be knowing about the artificial sweetener. It is also like in the market also you have got the sugar free is there, right? So that sugar free is nothing but your aspartame. It is a low calorie artificial sweetener. Now the problem with this is that even though it is not increasing the calorie, just like how the sugar consumption increases calorie, the problem with aspartame is that high consumption of this aspartame, like in your family also, if suppose someone is having the diabetes, they start taking the sugar free pills, right? Now the problem with sugar free pills is that the person start losing hearing capacity. Okay, the ability to hear clearly is lost if you continuously take the artificial sweetener. So this is not given over here, but this is the research has been done, and now many doctors are advising not to take the artificial sweetener. It is better to avoid sugar itself for diabetic patients. So this I'm telling from my own personal experience also. Then yeah, okay. So that's all about the amino acids. So, but it's not over, okay? It's not over. These are just the applications you can just go through and remember. So they are also used in pharmaceutical industries, for example, L-DOPA for the treatment of Parkinson disease and all, etc. So all these are the applications. You can just go through them. There's nothing to explain. You have to just by heart actually. Okay, so that was all about the amino acids, and now we are going to start with the from tomorrow onwards we are going to start about the proteins. Okay, so about their structure, Ramachandran plot, primary, secondary, tertiary, etc. So thank you for listening.